Developing biotechnology value chains that applicable locally is important to reach circular bioeconomy. Please welcome TALTECH Associate Professor, co-founder and co-CEO of IO company, Dr. Petri Jaan Lahtvee.
Hi, hello everyone. Indeed, I'm Petri Lahtvee. I'm an associate professor at the Tallinn University of Technology and also a co-founder of IO. And today I'm trying to tell a story how we have spun out a company out of the research that we have been doing at the university. I wanted to start with this graph over here. So it demonstrates the global overshoot days of different countries. And if you are not familiar with the context, then what it means is that it pictures if the whole humanity would behave or would consume in a similar fashion as the people from this specific country, then when we would deplete the resources of the world. And why I'm showing this is because just yesterday over here, it's about 43 days into this year, Qatar, if all the people, all the humanity would consume as people from Qatar, then our global resources would be depleted by now. And unfortunately, Estonia is not doing much better neither. So the predicted overshoot day for Estonia is March 8th. So pretty soon. This indicates that we really need to take some action here. Already today, the world is not enough. In average, we are using 1.7 Earths per year. And by 2050, the predictions are that we need 3 planets of Earth to sustain the human, our consumption requirements. And of course, as shown already in the previous graph, different countries consume differently. But we are doing actually worse than US today. So in Estonia, we would need more than 5 planets Earth to survive. And of course, in Estonia, we know the energy generation is pretty expensive and also polluting. But over here in this graph, it's from 2020, but it nicely shows how, between different sectors, how we are emitting CO2. So the largest parts are power, we have the transportation, we have buildings, we have industry, and agriculture. And this is how much CO2 we are emitting. And then we see the small bar below zero, which is LULUCF, so land use change, and forestry, which indicates how much we are fixing CO2 at the same time. And you can see that this is a pretty, it's a huge difference how much we are emitting and how much the Earth can actually fix the CO2. We also know that by 2050, European Union has a target to become net zero emitter, meaning that we need to bring down the CO2 emissions. And of course, also in this report that I'm referring to, they have the strategies how to do that. First of all, we need to reduce the emissions in the power energy generation sector, and then we can do it for the other parts as well. And it's doable, but we really have to invest into this. We have to come up with novel solutions and novel technologies to do that. This is not the only way, but the way how we have chosen to tackle this problem or this issue is re-engineering life. We are trying to apply biotechnology. We are redesigning microbes to solve or provide solutions to materials, fuels, and food that we are consuming today, so the processes would be more sustainable. And from the other hand, we want to use locally available sources or resources as a raw material for this process. So everything that is renewable, we could or should use as the raw material for those processes. And at TALTECH, my research group is called Food Tech and Bioengineering Lab. We are about 15 people there. We are collaborating with several other universities, also in Estonia. And we have several projects running at the moment where we are exactly tackling these kind of issues. So what we are doing, we are building biotechnology value chains. And what is the basis of this value chain is having a microbe that does all the work. So microbes you can find everywhere. They are in our gut, they are on our skins, they are in the air, they are inside the food that we are going to consume later during the lunch. They are doing a lot of different functions already naturally, but to make them do, or program them to do something that we really want to do, we need to engineer them. And therefore, we have developed tools to do that. And in our research group, we are working with several projects. So the furthest, we have gone with the production of bio-based coating materials. We are working in a consortia project where we are producing UV curable and water-soluble paints. And our goal is to replace at least 25% of fossil-based materials in coating materials with bio-based ones, but today we are already on a target to reach 75% of bio-based materials. And that means providing binders, fillers, pigments, and also solvents that are bio-based. It's not an easy task because we cannot provide a product that don't have the same properties that fossil-based ones have. So the consumers have used with a good quality. The paints have to be resistant. They cannot bleach and so forth. So it's actually a quite difficult task, but in collaboration with different partners and also companies who are testing these materials, we have come up with pretty good solutions already that are currently scaled up to pilot scale and tested in real use cases. Additionally, we have just started another project where we are producing drop-in chemicals for aviation fuels. It's a Marie Curie project, also a collaboration project with several universities in Europe. And currently we are looking for PhD students to join this project and start producing terpenes to add as a drop-in to aviation fuels. And also we are interested in producing plastics. It's a quite similar chemistry to binding materials in the coating materials. So providing monomers as a raw material for plastic production. From the other hand, we are looking more into the food and cosmetics solutions as well. We have an ongoing project where we are trying to replace fish feed. So today, fish feed mainly consists of bycatch from the seas, but it takes out a lot of fish from the oceans and contributes to the loss of biodiversity. So we are trying to recycle waste that we are creating to produce actually healthy food for fish. And also we are producing different types of lipids, fatty acids, and for example, astaxanthin that is used as an antioxidant and pigment ingredient in food and feed as well. And where do we get the raw material for all of those processes? So as mentioned, we want to use locally available resources. So first of all, in Estonia, it's important to use lignocellulosic biomass. So here we are collaborating with, for example, Fibonol, who is producing sugar hydrolysates. We have especially focused on hemicellulosic sugars because today there are not that many industrial applications for hemicellulosic sugars. But we are also working with Borregaard and also with, from Timo Kikas, we have received materials from agricultural waste, hydrolyzed agricultural waste. Additionally, we are looking into food waste. From one side, different side streams from food industry, but from the other hand, also the actual food waste. Today, in European Union, about 1/3 of food that is created are actually going directly into trash. And this is something that we want to tackle. It's not an easy task because the food gets spoiled very fast, so we have to do something very quickly with it. And in collaboration projects, we are today using anaerobic fermentation, known very well also for biogas production, but we are stopping the process before the methane is produced and using then short-chain fatty acids as a substrate again to feed those microbes and go towards these final products that we are interested in. And we are more and more ramping up the research on the process of using products that are coming from fixing CO2. So here also it's a collaboration with, for example, Kaspar from Tartu University, who is fixing CO2 into acetic acid and ethanol that we can further use. And there are other collaborations ongoing in this set of research as well. So several different raw materials that can be used for these bioprocesses and different products that can be produced here. So to facilitate this process as efficiently as possible, we have screened a lot of microbes which could do that. And we have stumbled upon yeast. It's a non-conventional yeast called Rhotorula toroloides, which has several positive characteristics to be used as a microbial cell factory to facilitate these biotechnology processes. One of its benefits, for sure, is that it can already naturally consume a variety of different carbon sources. Carbon from lignocellulosic hydrolysates, carbon from food waste, small molecules like acetic acid or ethanol. It can be easily and quite efficiently consumed by this microbe. And from the other hand, it already naturally produces high amounts of fatty acids or some interesting enzymes that can be commercially used as well. So we have adapted this organism, but as it's a non-conventional organism, it lacks its metabolic engineering tools, so this is something that we had to start with. And we came up with we took an engineering approach from, not from biological engineering, but from traditional engineering side, where you develop a design, build, test, learn cycle to upgrade the microbe in every cycle. So what we have done here is we have first developed tools to better understand the microbe, to look into its genome, to understand its proteomic and transcriptomic profiles, and based on that created metabolic models that guide us how to engineer the cells to make it more efficient for the cell factory applications. So using those models, we create a strategy how to engineer a cell. Then we go into the second phase, which is a building phase, so a genome engineering of those microbes. And for that, as mentioned, we have developed a CRISPR-based and the more traditional Golden Gate-based genetic engineering tools to manipulate the metabolism of this yeast. Next, when you have created new strains, you need to validate that whether these hypotheses that you had, whether they worked out or not. So therefore, we have a testing platform. We have lab-scale testing where we have 1 liter bioreactors and also high-throughput, smaller-scale reactors to observe the growth and the production of those strains. When this is done, you collect the data, you run the analytics, you understand how well they are producing something, how well the growth of the microbe has changed, or has it improved, has it reduced, and this data feeds into the computational models again to improve the models and provide new suggestions. And this strategy goes into an iterative loop until we reach to a strain that actually can produce something on a high scale that it would have also industrial relevance. And then what needs to be done is the process needs to be scaled up. So first you have tested everything in lab scale. You assume that everything works. And then, yeah, next step is to put it into closer to the industrial scale. And here we actually, in our research, we saw a missing link because running those scale-ups, it's quite expensive. It requires additional funding that you usually don't have in research projects. And initially what we tried to do is when we had reached this level where we felt that we have some strains that are quite good in their production. We were working with lipid production at that time. And we tried to convince some of our collaborators to take this idea and take it towards the industrial applications. We were not lucky with our partners. They didn't have the motivation to take on this project. So what happened then was that we actually, with my co-founder, we founded a company to do this scale up ourselves and apply for dedicated funding to actually do the scale ups. And as mentioned, the first products that we were producing were fatty acids. And why did we choose to go after the fatty acid production is that actually the market for the production of fats and oils is huge. It's about 235 billion euros yearly, or dollars. And quite large of this is actually produced unsustainably. So big part of the oils are produced with palm trees. Of course, palm trees are very efficient, but this process is not very sustainable. So therefore, especially where these unsustainable oils are going, what we saw was the food applications mainly. And when we are looking more deeply into the food applications, we can see that our current food system is broken. It's unsustainable. Most of our food is produced unsustainably. About 1/3 of all the greenhouse gas emissions today are coming from the food production. And quite often, the food that we are consuming is not even healthy for us. So we are quite often consuming quite a lot of meat, which is not actually good for our health neither. So main thing how to mitigate this issue is to come up with alternatives for animal-based meat. And today already we have quite a lot of options how to produce alternative proteins. And people are actually embracing these alternatives. There's predictions that the alternative meat market will grow to 1.1 trillion euros by 2050. And there are a lot of signs that people are adapting actually these flexitarian diets. So they're consuming less meat, replacing their animal-based meat products with alternative ones. But the main issue here is that still today, this food not always tastes really good. And one reason why this is happening is because these food products don't contain good fats. Because fats give the taste to our food, it gives a nice texture to the food. So fats are therefore a critical element here. And therefore, me and my co-founder 2 years back founded a company called IO, where we are producing future-shaping fats and oils. Our solution is to provide fats and oils from low-value biomass with a smaller economic footprint. And how we do that is actually the same biotechnology process, so brewing fats and oils. We are using sustainable raw materials. We are using fermentation process, using yeast, and producing fats and oils which are good for food, feed, cosmetics, applications, and also for household chemicals. So these are the 3 products that we are providing today at the company. We have solid fats, or we call them encapsulated oils. We have liquid oils, and we have our most interesting product, I would say, is the solid fats. So we are today the only ones who can provide solid fats with non-GM microbial strains. And if we are putting our technology into perspective, then what we can say is that we don't use any plants or animals to produce those fats. Our production is going under the circular economy value chains, meaning that we are using waste from other processes to actually produce food. And therefore, we are mitigating tons of CO2. So we have made calculations that replacing palm oil production with our technology, like replacing 1/3 of palm oil production with our technology, would mitigate about 100 megatons of CO2 per year. Also, what is important, I see that the slides are shifting somehow. Let me see if I can make it a bit better. No, I'm not a Mac person, so if you can maybe put it down again. So what we are showing, what we can say is that we are also contributing into food security, because we don't rely on long transportation chains, but we can produce food locally, or fats and oils locally, and also we don't contribute to the loss of biodiversity. Today at AYO, there's 11 of us, and our goal is that the tasty food would not be the opposite meaning for the healthy food. So today, quite often, when we are thinking about healthy food, we don't feel that it's tasty or something that we really want to consume. But our goal is that these two things would be the same thing, and all the food would be produced sustainably. Thank you for your attention.
Thank you, Petri. And now I see we have a lot of questions.
Thank you. Very nice talk. You mentioned that your mission is one of your missions is to replace the palm oil with your technology, and you want to produce it locally, and the market is huge. So let's say hypothetical situation that you have access to the all waste biomass, which is not from the field, which is not competing with agriculture, like not sugar beet-based biomass, and you use your technology. What's the fraction of the global palm oil, let's say palm oil, coconut oil combined? What's the fraction of this you can replace? And how many factories, the stainless steel and quite a footprint-rich factories you need to build to replace it all?
Yeah, it's a good question. We actually have done those calculations, and it's quite interesting. So we took that we took a goal that we want to replace 1/3 of today's palm oil production, and for that we need 7% of agricultural waste that is produced in Brazil today. So we took Brazil as an example because in Brazil there's a lot of agriculture, and my co-founder is Brazilian, so there's some connections as well. So 7% of agricultural waste created in Brazil, we can turn it into to replace 1/3 of palm oil. For that, we need about 230, I would say, average size factories. So it's more or less the same size as Saku or A. Le Coq brewery in Estonia. And as mentioned already, that we could actually, with that, we could already mitigate 100 megatons of CO2 emissions.
But Estonian biomass?
From Estonian biomass, well, in Estonia we don't need that much palm oil neither. So
what's the capacity you can, if you contribute to Estonian economy, and what's the scale you can actually operate in?
So we have considered that one factory should consume about 100,000 tons. 100,000, sorry, 100,000, tons, no, million tons, so megatons then, of product. So it's what is it? So if it converted now into the wood chips and sawdust, let's say, then in Estonia, we take down about 10 million cubic meters of wood. And we are considering with a factory that is then, 1,000-fold lower, that it requires 0.1%. And we can produce about, not cover the whole Estonian use with that, but about 1/3 of Estonian use of fat. So these are very tangible numbers, actually, we are operating with.
Yeah, thanks.
Thank you.
Yeah, thanks. I was first about to ask you about the genetical modifications. Why can't you use Golden Gate? But then I saw your paper on this. So that pilot plant picture that you showed, is that up and running in Tallinn? Is that your facility already?
No, this was not...
Oh, it's an illustration only.
It's an illustration,
right.
But actually we are building a pilot scale production at the moment.
So how large reactors?
So today we are building 300-liter facility, so it's very still small or like large lab scale, let's say. But next step, what we are planning and doing engineering already is for the demo scale.
We have 5 1,000-liter reactors lined up in Tartu, so if you want to test them, your stuff, we are eager to discuss this. But the actual question is about the food waste streams that you mentioned. I mean, we all know about all the difficulties of converting the wood material into glucose, right?
Yeah.
There are 1,000 lignocellulosic reasons for that not to work. So can you break it down a little bit, the numbers? How much this kind of a starch-rich or the carbohydrate-rich food waste streams are actually produced as a kind of a technological side streams in Estonia? And what kind?
I'm missing the numbers for Estonian context for that, because we are working on collaboration projects. So we are looking the whole Europe. And today we are working with actually agricultural waste. So we are not working directly still with the household waste, but in principle, this same technology should be possible to apply for household waste as well. But today, exactly, we are working with waste coming from agriculture, the fruits and vegetables which don't have a standard shape, or they have some kind of damage, and they are going just as animal feed or just as a waste. So we are currently working with those.
Yeah, because we grow a lot of potatoes, and if there is a viable food industry, for example, making chips, there's a potato peel, which is basically just pure starch, right?
Exactly.
Okay, anyway, we're over the time limit.
But one more question.
Okay, thank you. I try to make it quick. Question about chemistry. So you mentioned this artificial meat, and actually the fats and meat that are considered not very healthy. So what is your strategy in a way on sort of what structures do you chemically aim for in your approach?
Yeah, so it's a difficult question, because ideally we would like to avoid saturated fats as well. So one thing that we want to avoid, and what we are avoiding for sure, is also trans fats. So this is something that we have 0% of trans fats in our microbial oils. Also, our oils contain quite a high amount of omega-3 fatty acids, which is an additional health benefit. And then the question is about saturated fats. So this is a tricky part, because saturated fats, higher amount of saturated fats do make the fat solid, which is a desired property. So what we are doing is exactly balancing there, that we would be able to produce fats that have the desired properties, so they would be solid at room temperature or higher temperatures if needed, but from the other hand, still having the minimal amount of them. So it's the balance and trade-off there, of course.
Okay, thanks.
Okay, thank you, Petri, for your nice presentation. And now we are breaking for coffee break, for short one, till 12:15. And after coffee break, we will continue with second session by manufacturing from CO2.